Okay, right. We should be recording there. So let's cover eukaryote origins. We talked at the end of it about Lynn Margulis, and her idea was quite revolutionary at the time of uh, serial endosymbiosis. The idea is that eukaryotes gained an awful lot of their complexity, gained mitochondria and chloroplasts, and in her original idea, maybe cilia and flagella, although we don't think that's likely now. They gained it through a process of symbiotic relationships with bacteria. And we finished by saying that this turns out to be extremely common throughout the living world. It's all over the map. We're going to encounter this again and again. And there's a lot of single-celled eukaryotes that instead of, or in addition to the standard issue organelles, they contain bacterial endosymbionts that give them new capabilities. These are not identical to the first eukaryotes, of course. They've got 2 billion years of evolution behind them. But they might give us some idea of how endosymbioses might have looked in early eukaryotes, and they can give us some insights into how endosymbiosis can evolve. So, for example, that's an amoeba. Um, if anybody does not know, amoebas are eukaryotes with no fixed shape that move by what's called amoeboid locomotion. That's not helpful, basically blobbing from place to place. This particular one is common in pond mud. There's probably a bunch of them in Lake Conway, or there will be when it thaws. It's called Pelomyxa palustris. It can get up to 5 millimeters long. It doesn't have mitochondria, but it's packed full of bacteria that carry out respiration for it. They're still identifiable as bacteria. Um, Ciliates Paramecium aurelia. You might well have seen these before. We've got some on order, so you might see them again, although I don't know what the situation is with orders right now, given the closure of everything. But anyway, Paramecium aurelia, there are strains of it that are packed with symbiotic bacteria. And there's various varieties of bacteria. They're called kappa particles, mu particles, there's alpha particles, and things like that. And the symbionts produce toxins that kill other members of their species that don't have the same symbionts. So it's kind of interesting to know that even among the paramecium, we evidently have fraternity rivalries, where paramecium with pi, kappa, and alpha particles are deadly enemies of paramecium with sigma, phi, and epsilon particles. That pause was for all of you to laugh uproariously. So moving on. Um, as we'll see, there's a number of amoebas out there that actually have shells over part of their cells, and they're called testate amoebas. You'll see this again, but I'll say it now. Test, uh, from the Latin word testa, meaning a pot, and test is used for any sort of vaguely pot-shaped covering around a cell. So this is a testate amoeba. It's an amoeba in a pot. It's called Pollinella chromatophora, and it's got a couple of blue-green bacteria inside. They're those things that look like green sausages. And Pollinella itself is not thought to be particularly closely related to plants or to algae. But right there, I'm tracing them with a laser pointer. There's one, there's another, there's one, and there's another. Those are blue-green bacteria. They reproduce when the cell does and get passed on to daughter cells. So they're pretty much permanent inhabitants of Pollinella, and Pollinella is not otherwise closely related to plants or to green algae. So we think this is a completely independent gain of blue-green bacteria as photosynthetic symbionts. It happened in the distant ancestors of plants and algae, and it's happened in Pollinella. I don't know if they still do this in 1A or 1B, but they used to do a lab where you'd get termites and squeeze out drops of gut fluid, and termite guts are absolutely packed with symbiotic protists. And this is one of them. This is Trichonympha. It's a nice big protist that, I don't know, always makes me think of hippie Santa Claus because it's got lots of long flagella and also this kind of shakes when it laughs like a bowl full of jelly shape to it. And the interior of this guy is packed full of bacterial symbionts, and it's the symbionts inside the Trichonympha, inside the termite, that break down the wood. There's lots of wood particles down here in the posterior half of the cytoplasm. You can see them there. Can everybody see that laser pointer? I hope you can. There's a bunch down here in the posterior half of the cell on the right, and there's lots of bacteria on, check the chat. Okay, good. You can, I assume that means you can see the pointer. And there's lots of bacteria on the inside, and there's bacteria on the outside. These long filaments that you're seeing right there are not the cell's flagella. Those are bacteria stuck to the outside. And you find lots of crazy things like that in termite guts. The question of how a bacterium can go from being a parasite to being a symbiont, how can this sort of thing happen in the first place, was looked at beginning in 1966 by Dr. John at University of Tennessee. He worked with amoebas in the genus Amoeba. If our deliveries come through, you'll get to see some of these guys in lab. And he succeeded in infecting amoeba with a strain of bacteria. They were gram-negative rods. Gram-negative, if you don't know, refers to a particular type of staining you can do that's related with the type of cell wall that they've got. So his amoebas got infected with a bacterial strain, and most of them died. Dr. John carefully cultured the surviving amoebas. So he got a strain of amoebas that were permanently parasitized with these bacteria. And at first, he found that he could cure infected amoebas by giving them a dose of antibiotics. Any howls in the background? That's my cat. Well, one of them. Hello, cat. But he kept raising these amoebas in culture. And after about 5 years of, generation after generation of infected bacteria, infected amoebas, the surviving amoebas had become dependent on the bacteria. They had gone from being hosts of a parasite to symbionts and a mutualism. The amoebas could no longer survive without them. And if you gave them antibiotics, the amoebas wouldn't be cured, the amoebas would die. It turns out that the bacteria were now making a key enzyme that the infected amoebas had lost the ability to make. So that's a lab model for how those endosymbionts might have been acquired. It is possible, under the right circumstances, for a parasitic relationship to actually evolve into a mutualistic relationship. As we'll see, the details of the original symbiosis were rather different, but that at least gives you a how possibly answer to the question of how symbioses could arise in the first place. And this was something that Lynn Margulis herself was really big on. evolution by natural selection gets stereotyped as being survival of the fittest. And I hate when people say that, partly because Darwin himself never coined that phrase. That phrase was coined by a guy named Herbert Spencer, a British philosopher, probably one of the most boring people in history. But Darwin didn't actually say that. The other reason I hate the phrase survival of the fittest is everybody assumes that it means survival of the meanest, survival of the most savage, ruthless, bloodthirsty organisms, survival of the lion savagely tearing apart a helpless gazelle on the Serengeti plain, or a great white shark savagely tearing apart a helpless guppy in the ocean, or something like that. And there are people that argue, sometimes approvingly and sometimes not, that this is basically a mandate for humans to treat each other in the same way. But this is utterly and totally wrong, because cooperation, including symbiosis, is at least as important in evolution as competition, arguably quite a bit more so. Survival of the fittest has never meant survival of the biggest jerks. Very often it is the best cooperator, not the best competitor, that ultimately survives and gets to pass its genes on. Symbiotic relationships can very often be favored by natural selection. Symbiosis is a major source of diversity. It's a way for evolutionary change to happen relatively quickly, because all sorts of possibilities open up when you take two different organisms with two different genomes and put them together in close association. The resulting symbiosis has, capabilities that neither of the original organisms had. They can even end up genetically engineering each other, because you'll often get gene transfer from one organism to the other. And again, you see it all over the place. We'll come back to it again and again, looking at it in the animal kingdom. And I mentioned that gene transfer was a possibility. The reason why your mitochondria can't decide anymore to leave you and go off and live independent lives is that roughly 90% of their genome has been transferred to the nuclear genome. So 90% of your mitochondria's original free-living bacterial genome is now part of your chromosomes. You've been genetically engineered with bacterial genes. Sounds gross, but an important thing to remember is that, we can do a lot of crazy things with genetic engineering, but we're not really doing anything that organisms in the wild haven't been able to do. We've got lots of cases of lateral gene transfer between organisms that originally weren't related. It does happen in the wild. It happened before we were around. We're sort of hurrying up the process and finding some new ways of doing it, but in and of itself, there's nothing unnatural about blending DNA from different organisms. Mitochondrial sequences sometimes still transfer to host genomes, and you get these bits of mitochondrial DNA that don't code, that are called nummets, nuclear sequences of mitochondrial origin. At least 5 human genetic diseases are now known to be caused by nummets, and chloroplast genes have gotten transferred to plant nuclei. Arabidopsis is a dinky little mustard that's used a lot in genetic research. Oryza is rice. Both of those have been studied very intensively by geneticists, and they both have nearly complete plastid genomes inserted in their nuclear genomes. They've got almost all of the genes that their chloroplasts' ancestral blue-green bacteria would have originally had. About 18% of Arabidopsis genes evolved in blue-green bacteria. So yeah, we're all genetic chimeras to some extent. And we talked about the tree of life, and I pointed out that in some ways that's not an accurate model, because with symbiosis in play, branches of living things can come together and join together so tightly that you can hardly see where they've come together. But lateral gene transfer adds yet another wrinkle to it, and I think the best metaphor for large-scale evolution might be the remix of life. That your DNA is, in fact, a evolutionary, DJ Darwin's, I don't know the language here, uh, EDM, Dirty South, chopped and screwed dance remix, or something like that. Okay, my attempt to be cool I think I just heard my wife laughing. My attempt to be cool has ended in utter failure. So let's move on. It just makes for a much more complicated world, but one with a lot more potential for amazing evolutionary adaptation than in Willy Hennig world, where things evolve in a reconstructible branch tree fashion. Cat, that better not be. I hope my cat just farted. Right. And Margulis argued that endosymbiosis was far more important, far more fundamental than neo-Darwinism, by which she meant the standard model that's sometimes called modern synthesis evolution, where new alleles appear by mutation of existing alleles and then change in frequency from one generation to the next. This is sometimes dismissively called beanbag genetics. This is the population genetics that you might have learned. This is good old p squared plus 2pq plus q squared. That's Hardy-Weinberg equilibrium. And Margulis argued that endosymbiosis was far more important than that you couldn't reduce evolution to the neo-Darwinist modern synthesis. And as she wrote, neo-Darwinism must be dismissed as a minor 20th century religious sect within the sprawling religious persuasion of Anglo-Saxon biology. I'm not entirely sure what she meant by Anglo-Saxon biology. I've always done biology in modern English. It'd be kind of hard to do in Old English, but whatever. Neo-Darwinian language and conceptual structure itself ensures scientific failure. Major questions posed by zoologists cannot be answered from inside the neo-Darwinian straitjacket. I got to see her give a talk on this subject in a scientific meeting in '98 in Boston. It was the Society for Integrative and Comparative Biology. And she talked like this about the need to end neo-Darwinism. And she was so forceful about it, I thought she was going to start calling for the end of neo-Darwinists. And I was kind of waiting for like her grad students to burst into the hall wearing Chairman Mao caps and carrying AK-47s and start lining people up against the wall. Now, obviously that didn't happen because I'm still around to tell the tale. But she was very insistent on this point, to the point that I've had her quoted at me by creationists as evidence for the fact that all of this is BS and life began 6,000 years ago and Noah built the ark and all that. Most of us don't see it as a refutation of neo-Darwinism, but as an expansion of it. Darwinism itself, Darwin's idea of natural selection boiled down to its very simplest steps is that individuals vary, that variation can be inherited, and some of that variation makes a difference in organisms' odds of successful survival and reproduction. And sometimes that variation that's heritable might come about through mutation, but some of it can also come about through symbiosis. An organism, if we have a population of organisms and some have a symbiont and some don't, that's variation. That variation can be passed on, and that variation might make a difference in the organism's chances of survival. So, as one of the grand old men of neo Darwinism, John Maynard Smith, put it, there's no contradiction between Darwin's belief that adaptations arise by the natural selection of intermediates and the possibility that new evolutionary potentialities may arise suddenly if genetic material is brought together by symbiosis. So it's all good. Darwin's not going anywhere, but Margulis expanded our ideas of what was possible a great deal. And I actually designed this several years ago. Darwin gets the fish that you see on car bumpers, but here's the Margulis fish. The Darwin fish is still walking around, but there's the little Margulis symbiont inside that is definitely helping to make it go. All right, I'll take just a moment for all of you to laugh in delighted wonderment. Sorry, if you hear any howls, the cats are having a free-for-all in the room right now. I don't know why, they're just cats. Anyway, we have 7 of them in the house, by the way. 7 of our own little household symbionts, although I don't know if they're quite as beneficial as mitochondria. But anyway, a little more details. We're pretty sure now that mitochondria were only acquired once, and that would have been at the very beginning of the evolution of eukaryotes, roughly 2 billion years ago. We've got various lines of evidence as to when that happened, and I'll talk about them in a bit. There are some protists that don't have mitochondria as such, and this is one of them. This protist is the cause of a human disease. It's called Trichomonas vaginalis. I used to volunteer as a lab tech at the Berkeley Free Clinic when I was a grad student there. And we'd see this in vaginal samples every so often. This is the causative agent of so-called or trichomoniasis infections in the human vagina. Not deadly, but not very pleasant to have, at least as far as I could tell. And every so often I would see these swimming around under the microscope in a vaginal swab sample that I've been brought by the medic. And these are protists that don't have mitochondria. They're anaerobic. They can't live on your microscope slide for long because they're killed by oxygen. And these were thought to have branched off the eukaryote tree before eukaryotes acquired mitochondria. So these were thought to be very, very primitive eukaryotes. These were thought to represent maybe what the ancestors of eukaryotes looked like before they got mitochondria. But we now know that even eukaryotes that don't have mitochondria have mitochondrial genes in their nucleus. So they did have mitochondria at one time. Some genes from those mitochondria got passed to the nucleus, and even if the mitochondria themselves are no longer there, the genes are still there. Anaerobic eukaryotes have organelles that are modified mitochondria. This is a cross-section through Trichomonas, and there's these blobs here that are labeled capital H. There's one there, there's one there, there's one, there's one. Those are hydrogenosomes. They don't generate ATP through oxidative respiration anymore. They generate some ATP and they produce molecular hydrogen as a byproduct. So they don't do what mitochondria do, but we think that what they are is in fact modified mitochondria that have evolved to do something else, mitochondria that don't do oxidative respiration anymore. So Trichomonas doesn't have mitochondria, but we think it has descendants of mitochondria, and it did have mitochondria as shown by mitochondrial genes in the Trichomonas genome. So we now think that having mitochondria is as fundamental to being a eukaryote as having a nucleus is. And this hypothesis was published in 2014, the inside out hypothesis. And the idea is that basically all of, or an awful lot of the distinguishing eukaryote features evolve more or less in tandem. So we start up here at stage one, this thing called an eocyte. That would be basically a prokaryote, and a symbiont, shown up here in orange. And the prokaryote, the eocyte, and the symbiont stick together, and the prokaryote host attaches to the symbiote using blebs, sticking out from its cell body. And you can see that here in stage two. In cell three, the blobs have expanded. They're engulfing the protomitochondria. And looky, what we suddenly have is that eocyte, the main cell body, is now a nucleus, and it's turning into a nucleus here in stage four. And the projections, the blebs, have expanded. They're fusing together. By five, they are completely engulfing the mitochondria. And spaces between these blebs, looky, we suddenly have an endoplasmic reticulum here. And we have a complete cell membrane, an ER, mitochondria in the cytoplasm, and a nucleus in the center. So the idea here is that basically all of these key eukaryote features, the nucleus, mitochondria, and the endomembrane system, all evolved in tandem through a pretty simple series of steps. Can we find evidence that that actually did happen? Well, we do have fossils that are old enough, but so far nothing quite like this. But there are living archaea that form blebs and attach to bacterial symbionts. Now, a brief refresher, we used to place all prokaryotes into one single superkingdom, but we now know that there are two major lineages of prokaryotes, the bacteria themselves and the archaea, that are not closely related to each other at all. Many archaeans, although definitely not all of them, are noted for being extremophiles, meaning they can live in places like hot springs or acid streams or hydrothermal vents, these volcanic hot springs on the floor of the deep ocean, places like that. And archaeans and bacteria look outwardly similar under the microscope, but on the chemical level, they're very different. And archaea are now thought to be the closest relatives to eukaryotes, or at least to the eukaryote nuclei. And these are as an archaean, it's called Giganthalma carutense. And the central thing that looks like, I don't know what it looks like, Casper the Friendly Ghost, is the archaean, and it's got bacterial endosymbionts stuck to the outside, kind of like stage two up here. So something like this could be a model for early eukaryote evolution. It's not the ancestor of eukaryotes, that was a very long time ago, but it does show how a eukaryote ancestor could have functioned. We can do a little better than this. In 2015, news was published of discoveries of new lineages of archaea, and they're now called the Asgard archaea. I'm assuming you've all heard of Norse mythology, either from the TV show Vikings or maybe from the Thor series of movies. You know, Asgard is the home of the gods of the Vikings, including the mighty Thor and including Loki, the trickster god. And this is a deep sea hydrothermal vent located between Norway and Greenland. It's an area where superheated, chemical rich water is bursting out of the deep ocean floor. Now, the temperature of the deep ocean floor is about 4 Celsius. It's about as cold as water can get and stay liquid. And you have superheated volcanic water coming into contact with water at 4 Celsius. So the temperature gradient is really sharp. And at this particular place, we have these weird pinnacles made of precipitated minerals when that mineral-rich water hits the cold ocean. And somebody called this Loki's castle because it does look vaguely like the sort of castle that a trickster god might build for himself. And in 2015, it was announced that we discovered a completely new group of archaeans from samples taken from Loki's castle. And they were in fact called Loki archaea. And we now have a lot more of these. They're not all found only in the deep ocean. We found them in other habitats as well. And they've gotten named the Loki archaea, the Odin archaea, the Thor archaea, the Heimdall archaea. They've all gotten named after Norse gods, you know, which I kind of personally very strongly approve of. But anyway, here's the weird thing about Asgard archaeans. They are, okay, this slide is, I could not find a slide that would show this in smaller font, but this is a cladogram. And all of these are various lines of eukaryotes right here. These are genes in the eukaryote nucleus. Let me see, there's a plant up here. There's green algae over here, red algae. Let me see, okay. The animals are down here. These are various other protists, some of which we'll be encountering. And the closest relatives of all eukaryotes are these Asgard archaea, and specifically a group called the Heimdall archaeota. And these Asgard archaea, Loki archaeum and Thor archaeota and Odin archaeota and all that, we now know they've got genes that we used to think were only found in eukaryotes. We found genes for actin, one of the proteins that, among other things, it's evolved in muscle contraction, but it's a part of the eukaryote cytoskeleton. Tubulin, the protein that makes microtubules, also part of the eukaryote cytoskeleton and a major part of the motor that makes cilia and flagella work. That's in Asgard archaeans. Actin regulating proteins called profilins. Histones, right? The proteins that your DNA wraps around when, you know, especially when cell division is happening and those chromosomes supercoil on each other. Hope you remember that. Proteins involved in vesicle transport and vesicle trafficking. So these Asgard archaeans have genes for proteins that we used to think only eukaryotes had. DNA analyses consistently place the Asgard archaeans as the eukaryotes' closest relatives. And here's the weird thing. Up until fairly recently, we had no idea what these things look like. We knew about these Asgard Archaeans from DNA sequencing. You can basically haul up some mud from the deep ocean floor, run it through a PCR rig, and it will spit out all of these sequences. But we hadn't been able to culture any of these bacteria alive. A lot of them we still haven't been able to. So it would be cool if we knew what Loki or Loki Archaea looked like. And now we do. We have two Asgard Archaeans that have finally we've been able to culture them and grow them in the lab, study them directly, and looky. This is one called Loki Archaeum ossiferum, and it's all blebby. You have a blobby cell in the center, and then you have all of these extensions sticking out, which ought to remind you of O2 or even 3 in our model. Here in this picture, you can see in this diagram, well, this is an electron micrograph, but these things that have been stained orange right there in this picture, those are filaments of actin proteins, and actins support these extensions of the cell. The other one that we've cultured so far, called Prometheoarchaeum, because they couldn't just stick with Norse mythology and had to drag in the Greeks, Prometheoarchaeum syntrophicum also looks very blebby. You have the basic idea of a cell body that sends out all of these extensions, and tangled up in these extensions are symbionts. There's another archaean called methanogenium, and a sulfur-reducing true bacterium called Halodelsulfa vibrio. Now, these two symbionts are not related closely to the specific ones that we think gave rise to mitochondria, but still, an archaean with these cell extensions wrapping itself up in symbionts is getting awfully close to stages 2 or 3 right here. So this is not proof that this scenario is exactly how things did happen, but it's evidence that it can work because we can find living organisms that very closely resemble what these hypothetical intermediates would have looked like. These hypothetical intermediates could have functioned perfectly well because living things very much like those intermediates are living perfectly happily on the ocean floor right now. And this is still very new. Prometheoarchaeum was only published in 2020, and Loki Archaeum was only published last year, 2023. So this is still very, very cutting edge and very exciting that we're actually getting some evidence that bears on the origin of eukaryotes. Okay, so I'm assuming you're all just staring at your screens going, wow, because this is pretty darn awesome. Right? You've got your mics turned off, but I'm assuming everybody's going, wow. Okay. Evidently I didn't properly edit the text right here, but we don't yet know of any symbioses between Asgard Archaea and the alpha proteobacteria, which is the group where mitochondria came from. But then the study of Asgard Archaea is still very young. You know, those two, we've only gotten direct cultures of 2 of the Asgard Archaea starting in 2020. So this is still very new. But for a little bit more insight into what the ancestral mitochondria would have looked like, alpha proteobacteria includes a clade of bacteria that don't have cell walls. They're called the Rickettsiales. They are obligate parasites inside eukaryote cells. That's what they look like under a transmission electron microscope. These are these cell wall-less bacteria. They penetrate inside eukaryotes, and some rickettsias cause human disease. So typhus is one. Rocky Mountain fever is another one. I think those are both carried by the bites of typhus is carried by lice, and I think Rocky Mountain's fever is caused by ticks. So there. Sorry if that caption was a bit of a kerfuffle, but hopefully I got the point across. And some pretty recent analyses, and this is only 2006, but as far as I know, the result still holds up. These are analyses of very complete sets of DNA and protein data. All of these names up here are various bacteria. Hello, cat, get off me. Oh, this is very sweet. I've got a cat on my shoulder. Try to remember how to concentrate with a cat trying to stab me. Right. All of these names are various bacteria right here, except right here, these three. Here, I forgot, I can draw on this thing. Those are not bacteria, those are mitochondrial genomes. And mitochondrial genomes show the closest relationship to this group right here, which is the rickettsias. So the closest relatives in the bacterial world of mitochondria are the rickettsias. And so that gives us the best idea that we have yet of what ancestral mitochondria might have looked like. They're related to a group that already is known for penetrating into eukaryotic cells and sometimes causing disease, sometimes not. Yes, you're a very nice kitty cat. Nice kitty. Why now? Why do you have to jump on me right now? So there. So that's where we are with how those symbioses originated. And we don't have a full answer yet, but the lines of the story are getting clearer. And that's a very interesting time to be working in this field. So stepping out of the cutting edge back to 1765, how do we classify eukaryotes? Well, the scheme that we use today was worked out by Carl Linnaeus, a Swedish physician who was on a divine mission to classify everything, because if you could classify everything in the natural world, you would understand God's purpose. You could get inside God's head and see what God had been thinking at creation, at least as much as any human could understand God's mind. That's why Linnaeus was doing this. And he had originally classified all living things into two kingdoms, the Animalia and the Vegetabilia. Incidentally, his third kingdom was the Mineralia. That was eventually jettisoned. We don't classify minerals with Linnaeus's system anymore, but we used to. So we classified everything as animal, vegetable, or mineral. And Animalia were things that moved around and could eat. Plants were things that didn't move and just sat there and were green and made their own food and grew out of the soil. Okay, sounds easy enough. There's animals and there's plants. As light microscopes started getting better, though, it turned out that there were lots of organisms that didn't fit with either group very well. This guy named Ehrenberg, in the background right there, that's his microscope. And even though his microscopes were not as good as the ones we've got now, he made some really excellent observations of single-celled living things. And he was one of the first to demonstrate that single-celled critters that he called protozoa were complete organisms in their own right, that they had complexity, that just as we have organs, they had, important functional structures, the organelles. He analogized different organelles with different organs. So if you've ever heard that the nucleus is like the brain of the cell, that's basically Ehrenberg talking right there. And a lot of these single-celled organisms didn't fit that animal-plant dichotomy. Let me see what's come up on the chat. Did Ehrenberg coin the term protozoa? I think he might have, but I'd have to go back and look that up. I know he coined some of the terms for different protist groups that we've pretty much jettisoned now, but they were still being taught in my high school in the 80s. So his work did have quite a long influence. But I'd have to go up and I haven't read up on Ehrenberg for about 15 years. I need to go check that out. Right. So just by way of example, there's a photosynthetic organism on the left. It actually can be pretty common in pond water. I've spotted it in great numbers in the water from Beaver Fork Lake at some times of the year. It's green. It's got protozoa was coined by Goldfuss. Okay, thank you for setting me straight. I knew Ehrenberg coined some names that stayed in use for a long time. But anyway, euglena was treated as a plant because it's green and photosynthesizes, even though it does in fact swim and move around actively. There's a non-photosynthetic organism on the right called Astasia that was treated as an animal because it swims and it eats. It can take in food by osmosis, and I think it can also engulf larger organisms with a mouth-like structure at the top end. And Astasia was treated as an animal. Euglena was treated as a plant. But it turns out that if you take a bunch of euglena and you culture them in darkness for long enough, the chloroplasts die, the green color is lost, and they turn into Astasia. So these are basically the same organism. It's just that Astasia is the bleached, non-photosynthetic form of euglena. So do we have a plant or do we have an animal? Interesting question. And it turns out that you can't just lump together all single-celled eukaryotes, as some people proposed. On the right, we've got about a 5-micron ball-shaped single-celled protist with a kind of brownish green chloroplast in it. That's this thing that I'm touching with the laser pointer. It's got a pair of flagella, short and long, and it's called Ochromonus. On the right, that's a 30-foot bull kelp called Phaeocystus. Used to see these things wash up on Central California beaches every so often. You might have heard of the kelp forests of Central California, where you have these enormous brown seaweeds rising up sometimes 100 feet with all sorts of fish and other things living in and among them. And they don't look anything alike. And yet it turns out that they're remarkably similar in terms of cell structure and biochemistry. They use the same types of photosynthetic pigment, for example. Hello, you horrible cat. Go somewhere else. And now the cats are trying to kill each other. Right. Where was I? So clearly we can't stick with this animal-plant dichotomy. Ernst Haeckel, Darwin's big German fanboy, who did a lot of work as a good scientist in his own right on some of the earliest branches of the animal kingdom and also on protist groups, put everything that wasn't a plant or an animal into the kingdom Protista. And in Haeckel's time, he threw in the prokaryotes and also the fungi. An earlier dude by the name of Hogg had put these organisms into kingdom Protoctista, but nobody has ever heard of Hogg. Protist is what you hear, except Lynn Margulis, who did hear about Hogg, argued that the right name should be Protoctista because Hogg was first. Maybe this is correct, but this has never really caught on outside of Margulis's own writings and the writings of her circle. So if you do research on this, if you come across the term protoctist, that's Margulis's term for what just about everybody else calls protist. So now you know what that means if you see that word. A fellow named Chatton in 37 split the protista and, in fact, split all life into prokaryotes and eukaryotes. And then it was Carl Woise in the 1970s, working with some of the earliest attempts at things like DNA sequencing, that split the prokaryota into bacteria and archaea, creating the three domain scheme that you might have been taught. Archaea, bacteria, and eukaryota. And then in 69, a guy named Whitaker separates out the fungi as a separate kingdom of its own. If memory serves, when I was in high school, the textbook was still putting the fungi in with the plants, if memory serves. But, you know, public high school in Louisiana was not exactly on the cutting edge, although the teacher was quite good. Thank you, Ms. Williams, wherever you are. But that creates the five kingdoms scheme that we used to teach here, where you have the Monera is the prokaryotes, and the bacteria, fungi, the fungi, protista, plantae, and animalia are the four kingdoms of eukaryotes. I'm not sure what they're teaching now, but for a while, this was the standard classification that you might have learned in 1440 or 1441. The problem is that the protista, or the protoctista, if you are Lynn Margulis, is defined by things it doesn't have, not by what it does have. The definition of protista is eukaryotes that don't have embryonic development and don't have extensive cellular differentiation. And by cellular differentiation, I just mean different cell types. Your body contains roughly 200 types of cell that do different things. A simple animal like a sponge might have, 5, maybe 10 types of cells. And protists supposedly don't have that kind of extensive cellular differentiation, but they're held together based on what they're not. And this always reminds me of a friend of mine in high school who used to tell jokes like, what does a bald eagle and a brick wall have in common? Anybody know? Put it in the chat if you know. What do a bald eagle and a brick wall have in common? Okay, I'm assuming everybody's asleep right now. But the answer is, of course, that neither one can drive a stick shift. And there are two things we can learn from that joke. One is that my hometown has an awful lot of weird chemicals in the water. And the other thing is that grouping things together based on what they're not is not good practice. I mean, it's true that bald eagles and brick walls cannot drive a manual transmission car, but saying that they're similar based on that shared lack of ability is weird. You know, that really doesn't work for us. And the protista is also defined by what it doesn't have, and that means it includes the relatives of true animals, true plants, and fungi, and a whole lot of groups that don't resemble any of those at all. And as we defined it, it's a paraphyletic group. There's no critter that was a common ancestor of all the protists that was not also an ancestor of animals, plants, and fungi. Protists are eukaryotes except for the animals, the plants, and the fungi. This is a little out of date. There have been some revisions to this, but this cladogram makes the point pretty well. This was a cladogram based on a large DNA data set from, I guess, 14 years ago. And I know you can't easily see all of the names on it. That's not really important. But the animalia are in that yellow box right there. Those are all the DNA sequences that belong to the animals. The fungi are in the red box down there. Those are all the sequences that belong to fungi. The green plants are up here. Everything else is a protist. And you can see there are protist groups that are more closely related to animals than to anything else. There are protists that are more closely related to fungi than anything else. There are protists that are more closely related to plants than anything else. And there's a whole lot of groups that just go absolutely everywhere that are not particularly closely related to any of the major kingdoms. And protists can be very subject to convergent evolution. When I was in high school, we were putting all amoebas into a group called the Sarcodyna. And I think that is one of Ehrenberg's terms, since you brought it up. I think he came up with that. And sarcodynes were eukaryotes that didn't have a fixed shape and moved by oozing from place to place through what's called amoeboid locomotion. And these happen to be three different amoebas: nuclearia on the left, acanthamoeba in the middle, and niglaria on the right. Always loved niglaria. I did my undergraduate honors thesis was on niglaria a long time ago in a galaxy far, far away. 1991 wasn't that long ago. Please tell me it wasn't. Anyway, so these are all amoebas. They used to be grouped together, and it turns out they belong to very widely separated branches on the tree. Let me see. Nuclearia is closely related to fungi. I don't even know if it's on the cladogram, but it's going to be somewhere right about here. Here, let me get the pen. Nuclearia is going to be somewhere around there. Acanthamoeba, the one in the middle, is right there. Niglaria is right there. So we have these three organisms that are all amoebas. They all move the same way. They all look superficially alike, and yet they're on three very separate branches of the tree. So clearly we can't classify protists just by how they look, because convergent evolution is pretty rampant. Here's the way we do it now. This is the base classification came out in 2011, and in 2019 they released a revision of it. This is the way that we classify eukaryotes now. There's two huge supergroups. One of them is called diaphoreticales, and I don't know why they gave it that cute little K spelling in right there, but it's called the diaphoreticales. The second is the amorphia, and then a group called the excavata may not be monophyletic. It currently doesn't seem to group with either the diaphoreticales or the amorphia, and it may actually be two groups that are not that closely related to each other. We still don't really know where they go. If you took parasitology with me, we talked about excavata because there's a lot of parasitic protists in the excavata. It may not be a monophyletic group, and we're not quite sure where it belongs. So we've got two giant supergroups, and the excavata we're still not sure about. Diaphoreticales includes Haptista. I'll talk about those later. Archaeplastida is the lineage of protists that first evolved symbiosis with photosymbionts. So that is green algae, red algae, and green algae's descendants, the land plants. And then SAR is a very big group with a lot of important members that we'll be talking about later. SAR consists of straminopiles, which includes things like brown algae and golden algae and diatoms. Alveolata, which includes ciliated protists, dinoflagellates, and apicomplexans, which are some of the big parasites. And Rhizaria, which include Radiolaria and Foraminifera, which have long names but big fossil records, and we'll see those coming up. And then Amorphia includes the amoebas in the amoebozoa. The amoebozoa includes all the amoebas except for the amoebas that are not in the amoebozoa. That was not helpful at all. And a group called Opisthocanta, which includes both Animalia and fungi. Yeah, where animals, the kingdom that animals are most closely related to are fungi. You have more in common with a mushroom than you do with a tree. Don't quite know what you'll do with that fact, but there it is. And on Thursday, we're going to start marching through these and covering major groups in this scheme. I still have to figure out exactly what order I'm going to do it in. I'm leaving out a lot of obscure taxa. There's a whole lot of mostly little flagellates that don't fit into any of the supergroups and don't fit into any of the major groups, and we're not always sure exactly where they go. Most of that we're just not going to have time to cover. Most of that is still under revision, but the basic supergroup scheme seems to be pretty robust, and we will be using this as our framework for going further. And we will go further on Thursday. Hopefully we will be all together, and I'm going to stop the recording now.